everyone, this is Horn here, and I've got some notes for Chapter 36. This is going to be a long one, and I'm going to talk really fast, so hang on. We're going to describe the methods which a plant uses to transport material from the roots to the shoots and into the leaves. In our discussion today, we're going to mention the short and long distance movement of material. We will bring back the definition of water potential and how we can relate that to plants. We're going to talk about how root pressure, transpirational pull, cohesion, and adhesion are going to get material all the way from the root tips to the tip of the leaves. We'll talk about guard cells and how they control the size of the stomata. And the last few minutes are going to be on translocation, which is the movement of material through the phloem. We're talking short distance movement.
$1 million mystery.
I love announcements.
The student written.
Okay, let's see if we can get through the rest of this with no more announcements. So, short transport of material in a cell. The cell membrane is going to control the movement of material in and out of the cell. We know this because the membrane is selectively permeable. We also know that the membrane consists of transport proteins that are embedded in the membrane. These sometimes bind to solutes in the extracellular fluid and then release them in the intracellular fluid, and that's how some materials can get into a plant cell. The plant cells also have selective channels that will allow only certain substances through them. All of these methods that I've been talking about are all modes of passive transport. So no ATP is needed, no energy. They are simply moving from a high concentration to a low concentration. A proton pump is also a way to move materials over short distances. This is where ATP is used to pump hydrogen ions out of a cell. The concentration of the hydrogen ions is higher on the outside, which makes the inside negative compared to the exterior environment. And having that charge separation, there becomes stored energy that's used for cellular work. This is very common in the root cells to take in potassium. Water potential is a term that we used during our biochemistry units and our diffusion and osmosis lab. We know that in osmosis, the water moves from a hypotonic to a hypertonic area. Hypotonic means lots of water, little solute. Hypertonic is the opposite. Little water but lots of solute. In plants, physical pressure also impacts how much osmosis will occur. Therefore, the solute concentration and pressure are both measured by water potential, and I put the symbol for water potential there. Water will move from an area of high water potential to an area of low water potential. Think of it like this. If it has a high water potential, it means it has a lot of water. That's the high concentration. It's going to move to an area where there is a lower water potential. An aquaporin is a specific channel meant for moving water in or out of a cell. Water does not move freely through the lipid bilayer membrane of a cell. It actually moves through a very specific channel designed just for water, and that allows water to move in and out of a cell faster. It's still passive transport, doesn't need any energy, but it allows the movement down the concentration gradient very, very quickly. Moving water a long distance, so long distance transport of water does not happen by diffusion. And I have a spelling mistake there. Diffusion is too slow to move water from the root all the way up to the stem and the leaves. So bulk flow is going to be a movement that moves water over a long distance very quickly. Bulk flow is driven by pressure. And as water is lost through the leaves by transpiration, which transpiration is the evaporation of water through the leaves of the plant, the pressure is reduced and tension is created that actually pulls the water up to fill the space that there was water there before transpiration.
David Schuin, please come to.
The picture that you have on the next page, I'll actually explain that after I get the next few definitions down. You need to know these different pathways, symplast and epiplast. Symplast is a pathway that moves material, water, between the cytoplasms of the cell, and the epiplast pathway moves water between the cell walls of cells. I also put the word endodermis here because it's in the diagram, and you need to know that the endodermis is an inner layer of anything, and in this particular example, a plant, whereas the epidermis is the layer of cells that are on the outside. So you can see the symplast pathway is labeled in blue, so water moving in through the root hair and actually travels between the cell walls of the plants to get into the xylem, which is the vascular tissue that's in the inner part of the root, and that's going to carry the water up to the shoots. Or the epiplastic root, which is drawn in pink, and goes into the root hair and then travels through the cytoplasm of all of the cells. I think I just said that backwards. The symplast is when water moves through the cytoplasm, and the epiplastic route is when the water moves through the cell walls. There we go. I figured it out. When water has to move up a tree, some trees are hundreds of feet tall. So proton pumps, water potential, those little methods are not going to get water very far. Those will get water into the root, but it's not going to move it up very far. So there are three methods that are really going to add some height to where water can move. One is root pressure. Root pressure is the upward push of xylem sap. Xylem sap is, we're talking about water. It still only goes a few meters up. So this is not the major driving force of getting water all the way to the tips of the leaves. It works most at night when roots will continue to pump minerals and water into the steel. This will actually lower the water potential there, and then water will flow in from the cortex, which forces all of the fluid up. You might see evidence of this in the morning when you see a little droplet of water hanging off the tip of the leaf. That's because of root pressure that was happening overnight. Transpirational pull, this is going to get water up a significant distance in the plant. This is an upward pull. Root pressure was a push. Transpirational pull is an upward pull of xylem sap. It's going to move material from the roots all the way to the leaves. This happens when the air outside the leaf is very dry or has a lower water potential, and water vapor will exit through the leaf, through the stomata, and when that does that, it creates a negative pressure. It creates a negative pressure which pulls water upward. Cohesion and adhesion are terms that we did discuss when we did biochemistry. Cohesion is when water sticks to itself due to hydrogen bonding, so water molecules can stick to each other and kind of climb each other up a plant. The other term is adhesion, and adhesion is when water molecules stick to other things, and in this case, water molecules are sticking to the hydrophilic walls of the inside of the xylem. Hydrophilic, remember, means water-loving, so water can stick to it, can touch it with no problem, and the water will move up the xylem and overcome gravity. This picture is nice because it shows root pressure, transpirational pull, and cohesion and adhesion. The bottom here's showing a close-up view of root pressure, water coming in, and this white structure here is the xylem. And then as the water moves up, showing a portion of the stem, this is a picture that that demonstrates adhesion and cohesion. So if it's cohesion, that's when the water molecules are sticking together. Adhesion would be when the water molecules are sticking to the internal structure of the xylem. And then the upper picture is transpirational pull. It's showing water in the form of a gas leaving through the stomata, and as it moves out, more space is available for water to move back up. Guard cells. All plants need a mechanism to control how much water is lost. The amount of water in the perfect balance is a delicate thing to control because plants need water to carry on photosynthesis. So if not enough is there, they can't carry out the process, and then they can't make any sugar. So a guard cell's purpose is to help balance the plant's need to conserve water with the requirement for photosynthesis. And I have a picture here that shows the guard cells and the stomata. The guard cells are these cells on the outside, and there's 2 guard cells for every stomata. Stomata is the opening on the inside. In this view here, this shows the guard cells being turgid, T-U-R-G-I-D. Turgid means full of water. They're swollen, and because they're so full, it opens the space in between them, so the stomata is open for business, open for oxygen to be released, open for carbon dioxide to come in, open for water vapor to leave. So photosynthesis is happening very quickly when the stomata is open because gases can come and go as needed. When the guard cells are flaccid, F-L-A-C-C-I-D, flaccid means they have shrunk. They are not full of water. And when they do that, it closes that opening, and it might close it completely, or it might make the opening very small. This might happen in times of drought when not a lot of water is available, and the plant is actually trying to conserve the gases and the water that's inside the plant. The bottom here are a couple things I've already mentioned. When guard cells take in water, they become turgid and swell, and this is when the stomata widens. When the guard cells lose water, they become flaccid and the stomata closes. The stomata is typically open during the day. So for the most part, most plants have a stomata open during the day and they close them at night. By closing it at night, it prevents any unnecessary loss of water when photosynthesis is not happening. The stomata can also be activated by a couple of different things. So not only is it open during the day and closed at night, but there are other factors that contribute to the opening and closing of it. They are activated by light. So as soon as the sun comes up, they will open. If there's a decrease in carbon dioxide gas in the leaf, which means photosynthesis is going to decrease, the stomata will close. And the purpose of that is to conserve the amount of water and carbon dioxide already in the plant. And the other thing that controls it is its internal clock. And this is called a circadian rhythm. This is something that we'll talk about later in our plant unit in more detail. A xerophyte is a group of plants that are adapted to very dry climates. Plants like cactus and succulent plants, they have certain modifications that help reduce the amount of water that leaves through transpiration. So they might keep their stomata closed most of the day and open it at night. But that's a specific adaptation to those plants that live in very dry climates. And the last section I have here is on translocation, and this is the movement of food through the phloem. Everything up until this point has been about xylem and water and moving water from the roots to the shoots, and now in the last few minutes we're going to talk about food and glucose that's made in a leaf and how it's going to get to other places of the plant that need it. Phloem sap. Phloem sap is made up of a sugar, some minerals, and maybe some amino acids. In order to move sugar around, you have to have a spot where the sugar is made, and then you have to have a spot where the sugar is going to get dumped. The spot where the sugar is made is called the sugar source. I'm going to show you a picture in a minute. But this is the organ of the plant where sugar is produced, which that's where photosynthesis happens. And the best, most common area is a mature leaf. Now, where does it get dumped? We call that a sugar sink. This is an organ in the plant that needs sugar. A spot that's has lots of cell division going on in the growing roots, in new stems, in the fruit. If there's a fruit on the plant, that desperately needs lots of sugar to continue surviving. These sieve tube cells, which are cells that we mentioned earlier in chapter 35, these are cells that will carry the sugar source to the sugar sink. And here is the picture. This food is moved at about a rate of 1 meter per hour, and it's going to move by bulk flow, which is moved by pressure, which we talked about when we talked about the xylem. And in my picture here, here's the source. The shadow behind it is showing the leaf, which is implying that the source of this is in a mature leaf. And the sugar is then going to get put into this sieve tube, which a sieve tube is a conducting cell, as we've learned in chapter 35. And it will conduct it down to the sugar sink, which the shadow behind it looks like some kind of a fruit or a vegetable behind there. And it's going to get dumped wherever the sugar is needed the most, which could be a growing part of the plant or the fruit. So this gets us through all of the notes for chapter 36, and we're going to be using all this information in a hands-on activity in class, so I hope this helps.